In 1956, it was Muller who coined a term called as the phytoalexin, which is called as a phytoalexin. A phytoalexin is basically a kind of a chemical which is a post-infectional chemical in the plant. That is, upon infection, the plant produces a chemical that is called as a phytoalexin, which is basically involved in the defense mechanism. In 1963, there was a significant contribution which was made by J.E. van der Planck. Van der Planck, and he was the first person to found out the so-called as the vertical and horizontal resistance. Vertical and the concept of a vertical and a horizontal resistance in the plant against the pathogen. And he has also worked extensively on the epidemiology of the plant diseases. Hence, he is regarded as the father of plant disease epidemiology. The father of the plant disease epidemiology. So, but his significant contribution include the vertical and horizontal resistance in plants. In 1966, two scientists that were named as Van Schemling and Marshall Kulka, where they found about the use of systemic fungicides, the use of the systemic fungicides, basically, especially in particular, the oxetine compounds. Oxetine compounds, especially the carboxyl and oxycarboxyl. So this is considered to be as the first systemic fungicides that was developed and being used for the plant diseases. In the same way, in 1966, Williams and his co-workers from Australia, so they showed that the artificial culturing of the fungus that is called as the black stem rust, that is in case of wheat. Why it is so important that he contributed for the artificial culturing of this particular fungus, because this fungus is an biotroph, which is often not able to grow on an artificial medium, but on an oxenic culture, which is called as an oxenic culture. Williams and his co-workers, they were able to culture on an artificial medium, but it is not a very popular, but still they had able to grow on it. In 1970, Garrett, he worked on so-called as the biological control of plant diseases, the biological control of the plant diseases. And this was the first mainly on the biocontrol and mainly on the root diseases. In 1972, G. Rangaswamy, so he published a very popular book, I can say, where it was called as Diseases of Crop Plants in India. This is even today, this book has been very much popularly used. And so he was also a pioneer in bacterial plant pathology and also agriculture microbiology. And Basically, he was basically a microbiologist, but turned into a plant pathologist. In 1971, another scientist from India called as Dastur, so he was the first Indian plant pathologist to establish a species. That is, he was the first person to show the species that is called as Phytophthora parasitic. First publication or the first identification of a particular fungus causing the plant diseases. And another significant contribution in the context of Indian plant diseases, which is K.C. Mehta. So this is an often asked question: who is a scientist who has worked on the disease cycle of wheat rust in India? So it is basically the K.C. Mehta, and he was also responsible for so-called as the identification of the Puccinia path in India. That is, Puccinia is again a stem rust. He basically identified how this pathogen moves from South India to North India. That is called as a Puccinia path, how the pattern of movement of this particular disease or a pathogen. And in the same way, another person called as Sadashivam, who worked on the mechanism of cotton wilt disease. The mechanism, how the infection takes place, is the one which he was able to identify. And Tirumalachar is another MJ Tirumalachar, very important contribution. He was the first person to introduce the antibiotics, especially in plant diseases. That is, in the management of a plant disease. In the management of a plant disease. Very significant contribution. There is use of antibiotics in plant diseases. And as I was mentioning in the very beginning, R.S. Singh, who has written a book about the plant diseases, he has also worked on a disease called as a pythium, which is often known to cause damping of disease. Often known to cause a damping of disease. Okay? And Y.L. Nene, he passed away, but his significant contribution include his identification of a disease called as Khaira disease in paddy. Khaira disease in paddy. That is a zinc deficiency. Often it is a zinc deficiency. He called it as a Khaira disease. And he has published a book, which is a very famous book that is called as Fungicides in Plant Disease Control. Fungicides in Plant Disease Control. It is a very voluminous book which talks about different fungicides used and its mechanism. And it's a very useful book for anyone to refer about fungicides. Yes, Nagarajan, again, he has published a book in plant disease epidemiology. And basically, he has also worked on epidemiology of the stem rust and as well as the mechanism of resistance in the plant. S.P. Mathur and Neergard. So these two people worked on the seed pathology. Formerly, they were the directors of the Danish Seed Health Laboratory, the Danish Seed Health Laboratory, and it is still very popular. And Neergard is also the prior director of this particular institute. Okay. So these are all the contributions of several scientists who have worked on the plant pathology and the plant diseases as such. In the same way, there is another field called as the plant bacteriology, that is the diseases caused by bacteria. I did not talk about the contributions which started with Louis Pasteur, Robert Koch, who proved that anthrax disease of cattle, and Robert Koch, who was called who postulated the Koch postulates and established the pure culture technique and disproved the theory of spontaneous generation theory of diseases and proposed the germ theory instead in relation to diseases in humans and animals. When it comes to the plant diseases in bacteria, Thomas J. Burrell, so he was an American plant pathologist, and he was the first person to prove the fire blight of apple. The disease called the fire blight of apple and pear, and he was the first person to show that it was caused by a bacteria or a bacterium. However, he could not prove it with an experimentation. It was proved by E.F. Smith, Erwin E.F. Smith, who gave final proof, that is, of the bacteria causing the plant diseases, especially the fire blight of apple. He also worked on the wilt of cucurbits, and also another important disease that is called the crown gall disease. The crown gall disease. And later, it was in 1910, it was Chilton and his co-workers. They demonstrated that the crown gall disease, the crown gall bacterium transforms into the plant and produces tumors. That is, the bacteria enters into the plant and causes the tumor cells because of a tumor-inducing plasmid that is called as a TI plasmid. A TI plasmid. Later in the same year, Jensen, he was a person where he showed that the crown gall is similar to that of your human cancer. That is, in the same way how the crown gall causes a disease in plants, he related that event to the cancer in case of animals or humans. 1952, a very, again, significant contribution by J. Lidderberg. He coined a term called as a plasmid. Coined a term called as a plasmid. And in the same year, Waxman, so he discovered a wonderful antibiotic that is called as a streptomycin. Even today, it's being used, and for which he received the Nobel Prize. And the streptomycin is being now used in agriculture for control of the plant diseases. And in the same year again, 1952, Zinder and Lidderberg they discovered a process of a phenomenon that's called a transduction. The transduction is basically a process how the bacteria transfers the genes from one to another by the way of the bacteriophages. And in 1962, Paul discovered another bacteria that is called as a Delovibrio. This is again a gram-negative bacteria. A Delovibrio is a bacteria which normally kills another bacteria, so which is basically a parasitic bacteria, I can say. In 1972, New and Care, Alan Care, Alan Care, they used a biocontrol strain called as Agrobacterium radiobacter. The strain name is called as K84. This is a bacteriocin producing bacteria. This was used to control the crown gall disease. This was used to control the crown gall disease. In the same year, Windsor and Black, they observed a new kind of phloem inhibiting bacteria. A phloem inhibiting bacteria causing a disease called as the clover club leaf. A clover club leaf disease. And in 1974, Zenin, he demonstrated the presence of TI plasmid in Agrobacterium tumefaciens. So this was the first time where plasmid is transferred into a plant. This was a demonstration that was done. In 1980, it was Dye et al. He introduced a system called as the pathology. This I will tell you what is a pathology means. Normally, it is used for subspecies identification of a bacteria. A pathology is a subspecies of a bacterial species. In 1928, there was one Griffith who proposed the principle of transformation, which was further showed by Avery, McLeod, and McCarthy. There is with a simple experimentation that the principle of the pathogenesis or the transformation of a gene by using two mice. So later it was Avery, McLeod, and McCarthy showed that it is the gene that is transforming and giving a new character for any particular pathogen. In specific to Indian condition, M.K. Patel, he worked on a disease called as Xanthomonas, a particular Xanthomonas campestris. PV refers to Pathavar, a disease in Ipomoea, or in case of the sweet potato. And several other people also worked on that, but let us just stick to this one as such. If we talk about another discipline in plant diseases, it is the plant virology. That is study of the viral diseases in plants. There were some contributions which are similar to that of your animal and others, but in specific, some of them are specifically worked on plant viruses. Adolf Meyer was first to describe a disease of tobacco, which he called as mosaic Kranheat. This was basically a German name that is called as a tobacco mosaic. A tobacco mosaic. And he also demonstrated that this disease, that is a tobacco mosaic, is sap transmitted. It is a sap transmitter. In 1892, it was Dmitry Ivanovsky. He was the person who demonstrated that the causal agent, so-called again the causal agent, is the tobacco mosaic virus, which is oftenly called as TMV. Is oftenly called as a TMV, and he demonstrated that the TMV can pass through bacterial filters. The bacterial filters. So indicating what is that the viral particles are smaller than your bacterial cells. So that means it can be easily passed through the bacterial filters. And again in 1895, Smith, who showed about a disease called as peach yellows, the peach yellows, and he proved that this peach yellows is a contagious means you know very well now what is contagious mean, like COVID-19 or a coronavirus is a contagious virus. So he at the time showed that this peach yellow disease is a contagious disease. In 1898, Bejering, a Dutch microbiologist, basically, he was a microbiologist and a founder of virology. Basically, he was called as a founder of virology. And he showed that again, this TMV, whatever we call the tobacco mosaic virus, is not a microorganism. And he called it as a contagium vivum fluidum. That is an infectious living liquid or a fluid. And he gave a word that is called as virus. The first time that was given as a virus, which is derived from a Latin word called as a poison. Which is called as a poison. Then in 1929, Holmes, he provided a tool by which a virus could be measured by showing that the amount of virus present in a plant sample, that is during the preparation, is proportional to the lesion on a leaf. For example, if you have a particular leaf which is infected by a, let us say, TMV, okay, in a tobacco, so he indicated that the amount of particles present in this is equivalent to the size of the lesion on an appropriate host. Again, it should be in an appropriate host, and it is a contaminated sap. It is a contaminated sap. In 1935, W.M. Stanley's very good contribution, I can say, crystallization of TMV, for which he got a Nobel Prize. He got a Nobel Prize for crystallization of TMV. 1936, born and Perry. They proved that this crystallization of the crystalline protein or the crystalline the TMV consists of a protein and a nucleic acid. That is, the crystalline nature of the virus contains the nucleic acid and a protein. And I think it was in 1939, Cauchy and his co-workers, the first time the TMV particles or the TMV virus in an electron microscope. That was the first time where people were starting to observe. 1956, Garer and Scram they proved that basically the nucleic acid of the virus, fraction of the virus, is the infectious agent. That is, you know very well that the virus contains the so-called as the protein and the nucleic acid. So of which the nucleic acid is the infectious agent in the plants, both in humans, it is also the same. Later in 1966, it was Casanis who discovered a type of virus called as a satellite virus. This we will discuss what is a satellite virus in coming days. And at last, in this particular one, 1971, it was Daniel who first time showed about a type of virus, but without a protein coat, that is viroid, which is smaller than the virus, having around 240 to 400 bases. Basically, it consists of RNA. Most often it is an RNA. And the disease which was first identified was called as a potato spindle tuber viroid. This is the first one to be identified as a viroid disease. And in the same year, the Peter Perryman and Eva Hull so they first time showed ELISA, that is your enzyme-linked immunosorbent assay, ELISA. They first time demonstrated for the usage of identification and various other usages in the molecular biology. Coming to another discipline, that is the plant nematology. Plant nematology, which is again one of the subjects in plant pathology. Here it was nowadays this is a very serious problem. 1873, John Needham first time reported a disease called as a wheat gall disease. A wheat gall which is associated with the nematode. That is the wheat gall disease, which is caused by Angina tretici. Angina tretici. So later was Atkinson in 1892. Atkinson in 1892, who reported a disease called as root knot disease. This is a very common disease among all the vegetables. Atkinson showed that the root knot is associated with another disease, fungal disease, that is called as a Fusarium wilt. So both together they are associated and they can cause a serious problem in cotton. And 1913 was Cobb. So he had a very good significant contributions, mainly for the isolation of a nematode, which is often called as a Cobb's sieve method. Cobb's sieve method of isolation. And he was also involved in the taxonomy, that is identification and naming of the nematodes. At the last, it is in 1958, sorry. In 1958, Hammett. So he discovered about the nematode as the vector of plant disease. Vector is basically like your mosquito, which carries the dengue and other things. So there is, in plant diseases, there are several vectors, including your insect vectors. Hammett was the first person to show that the nematodes can also act as a vectors. Okay. And another small diseases, but now gaining a lot significance because of its importance, that is phytoplasmas, which were earlier called as mycoplasma-like organisms, MLOs, mycoplasma-like organisms. This was recently identified by Doyle and his co-workers, and Ishii also worked on it later from Japan. They found that it is a mycoplasmatic organism, mainly causing yellow type of diseases. That is, anything yellow on the plant is becoming yellow, was considered to be as mycoplasma-like organisms, and now it is called as a phytoplasma. So this, they found that the tetracycline can inhibit the phytoplasmas, indicating that it is one of the antibiotics that could be used to control. And in the same way, pyroplasmas also causing some of the diseases. And it was first time identified in 1972 by Davis and his co-workers, where they observed a helical wall-less microorganism causing a disease called as a corn stunt disease. Okay. This was the first time that it's been observed. So this is basically to have a brief understanding about the contributions made by different workers, which is a very significant one. If you look into all this, is very, very important. When we talk about the development of plant pathology, teaching as a plant pathology, the teaching as a plant pathology course was started in the University of Calcutta. That is, as a subject syllabus, and also in Madras. Go back in 1857. That's a long time back. Emphasizing mainly on the fungal taxonomy, because fungal are the major ones known to cause the plant diseases. But plant pathology as a university science was started in 1930 by University of Allahabad. University of Allahabad. That is and also in Lucknow. And as a course, University of Madras introduced the plant pathology, and later it was the Agra University introduced the postgraduate program in plant pathology in Government College, Kanpur. That was in 1945. But the organized way of teaching of plant pathology was started in IARI, that is Indian Institute of Agriculture Research. And the subject received an importance with supporting of all these disciplines, so-called as the mycology, bacteriology, the virology, nematology. So all these subjects together as combined and made into a plant pathology. And now, of course, all the agriculture universities do have the subjects, especially on covering all these fungal, bacterial, viral, and so on. This is basically to make you understand the importance of the plant diseases and its relevance with respect to the contributions many people made, which is really an important, I can say. It is never been ignored. It can never be ignored. Okay. So now we will get into the actual one. What are the causes of plant diseases? The causes of the plant diseases. We always say it is a pathogen. That is, when we call this COVID as pathogen. And remember that pathogen should be always associated with a disease. The pathogen should be always associated with a disease. What is pathogen? Pathogen can be any agent or it is a reagent or a factor that incites as pathos. Incites as a pathos or a disease in an organism. It could be a human, it could be a plant, it could be anything for that. So pathogen can be any agent or a factor that incites pathos or a disease is in an organism. So what exactly the pathogen means? It could be a microorganism or it could be a virus particle. And remember that it should be a transmissible. Okay, it should be a transmissible, which is often we call this as a biotic agent. We call this as a biotic agent. It's a biologically that can cause a disease in the plants by disturbing the metabolism. By disturbing the metabolism of the plant cells. How it does? It is done by either by the production of the enzymes or the toxins or the growth regulators. Or the growth regulators or any other substances secreted and which is responsible to absorb the nutrition from the plant. Absorb the nutrition from the plant's host cells. So it should be a transmissible biotic agent causing a disease by disturbing the metabolism through enzymes, toxins, growth regulators, or any other substances which can absorb the nutrition. So basically, if you see the plant pathogens are grouped as one is biotic, or it is the infectious. Biotic is basically a biological agent, or it should be an infectious to cause a disease. So here under this, we have the diseases caused by fungi, diseases caused by prokaryotes. That is, prokaryotes include your bacteria and molecules. Molecules mainly refer to your phytoplasma and spiroplasmas. And also the higher plants. Higher plants, this we call this as a Fenergonic plant parasites. Fenergonic plant parasites. For example, the phenerogamic plant parasites include, number one, is a total stem parasite. The stem parasite, which can be, the best example is Cuscuta, the total stem parasite. And we have a total root parasite, and there is your Orobanche. And the third one, we have a partial stem parasite, which is the Loranthus. I think this you might have seen in all these orchards where on guava and other plants, they grow on the top. And finally, we have a partial root parasite, which is the striga. In case of sorghum, it is a common parasite. Basically, these are plants which are parasitizing on the other plants. So you can just remember as coals: Cuscuta, Orobanche, Loranthus, and striga. Total stem parasite, total root parasite, partial stem parasite, and partial root parasites. And also we have the diseases that are caused by the protozoans. Though they are very less, we have some of the examples like heart rot of coconut and palms, and also the phloem necrosis of coffee. The phloem necrosis of coffee. There are also diseases caused by algae. This, if you go to any mango orchard or anything, you will see that there is some kind of a moldy growth on the leaves. That is nothing but your red rust of mango. Red rust of mango is one of the classical example, and in GKVK you can see quite a good infections on mango and even other coffee and other plants. Metazoan animals, and here we have the nematodes. And the second group of plant pathogens group is the mesobiotic. Meso means it is in between living and dead, because viruses, when they are in the host, they behave like a living organism. When they are in a living organism, so basically the examples are virus having nucleic acid plus protein, nucleic acid plus protein. And the best examples for us in case of this mesobiotic, including the viruses, the TMV, which is a common examples, and the tomato leaf curl virus is another example where we can put it under the mesobiotic group. And in the same mesobiotic group, we have the viroids. Already I mentioned about it, and the potato spindle tuber virus. Which is one again where you can say this is a naked without a protein coat, but this is also known to cause in some of them. Coming to the third grouping, that is what we call it as an abiotic causes, or which is oftenly we call this as a non-infections, because these are not biologically active. These are all, this could be, for example, if you have to take some of the examples of this kind, is like example, the low temperature. The low temperature is like, for example, the freeze injuries, especially in the temperate region. Normally, these freeze injuries are very, very common. And again, if you see, the high temperature is also responsible for as an abiotic. The example for this would be like heat canker of flax. The heat canker of the flax. And in certain cases, the unfavorable oxygen also can be an abiotic factor. Unfavorable oxygen, that is, if there is an imbalance in the oxygen level, normally it also can cause a kind of disease, especially a disease called as a black heart of potato.